and we'll get started. So I'm Reza Shadmehr, and thanks for being with me today. I'm going to talk about the cerebellum and its computations during control of movements. Now, those of you who know me, for the first 20-some years of my career, we had a laboratory that used computation to study human motor control. And the last 5-6 years, with the help of these wonderful, magnificent, really students, we made a transition to marmosets and studying the motor system in these animals. So I just want to briefly introduce them. Ehsan Sedaqatnejad was my first PhD student who worked exclusively on marmosets. Then Paul Hage, Jay Pai, and Amin Fakharian are current PhD students that have contributed to the work that I will show you in this presentation. So I'm going to talk about the cerebellum. And last week I was out on a little hike, and I did a little experiment that I wanted to show you. So what I did is that I took the cell phone and I sort of put it on my forehead, and this is what happened. So obviously, when I'm walking, I don't see the world this way, but somehow my brain is able to send some prediction signals that compensate for the movements that are taking place, allowing me to have this stable visual field. I'll show you another experiment to demonstrate this idea of stability. Here's some classic work that we did about 20 years ago. So you see that it's quite difficult to respond to a perturbation when it's induced by somebody else. But if you go to pick up the book yourself, there really is this rather exquisite ability to precisely turn off the muscles of the biceps, in this case, at the right time by the right amount so that the arm remains stable. And so, the idea is that our body is subject to things that occur, perturbations and so forth. We have a time delay reflex system that responds to it. But if we could make predictions based on the movements that we ourselves are generating, if our motor commands are things that can be listened to and make predictions based on them, then we can make compensation, as you saw with the stability of the hand. And for many decades, the idea has been that the cerebellum has something to do with this. And in robotics, this is called forward models. Forward models are these things that make predictions about consequence of these commands. And the question has been, well, is there such a thing, and where it might be, and does the cerebellum have something to do with it? So I'm going to be talking about the cerebellum, but the cerebellum is a rich field that began about 2 centuries ago with the observations of Pierre Flourens, who did lesion experiments on pigeons. And what he said was that, look, the slightest disturbance of the cerebellum altered the harmony of the coordinated movements. And this word harmony had a specific meaning to him. He compared the animal's movements, the pigeon's gait, to that of some person who had drank alcohol. And he wrote that the will, the senses, the perception remained, the power of movement was retained, but the coordination, the ability for controlled and determined movement was lost. So we don't need the cerebellum to make movements, but we needed to do these movements well. Ramon y Cajal, when he was studying the anatomy of the brain, when he looked at the slices of the cerebellum, what he realized was basically the individual neurons make up the fundamental computational units, and that became the neuron doctrine. It was based on his analysis of Purkinje cells. Gordon Holmes, a few decades later, Gordon was a neurologist who during the World War I and aftermath of World War I, studied the soldiers, the British soldiers that had returned from the war. And I want to show you what he looked at. So on the right here, what you see is the British soldier. And you see the helmets of the British soldiers have this opening in the back, whereas the German soldiers have this much better design helmet that covers their entire cerebral cortex and the cerebellum. And so, as you can imagine, this design flaw in the British helmet resulted in a whole bunch of British soldiers having gunshot wounds to the cerebellum. And so when he examined those soldiers, what he noticed was these dysmetric movements. The most obvious errors in the movements that they made were seen toward the end of the movement, as they were supposed to stop the movement on the target. David Marr was a theoretical neuroscientist that just by thinking came up with this remarkable idea that the cerebellum might be a learning machine. He proposed that the inputs to the Purkinje cells had synaptic weights that were modifiable. The inferior olive was the teacher of the cerebellum, and the Purkinje cells were the students. And this is really remarkable because, he just through thinking came up with this idea of learning. And then Masao Ito discovered that the Purkinje cells, which are the output of the cerebellar cortex, are inhibitory neurons, which is rather remarkable for an output structure. And the input from the olive produced learning in the parallel fibers, the inputs to the Purkinje cells. So there are really two ideas that I want to highlight here. There's ideas about control, which is the study of cerebellar patients and Gordon Holmes, and there's ideas about learning, which have to do with how error changes the cerebellum. And as I will show you, these two ideas, they really need to be combined for us to make sense of the neurophysiology of the cerebellum. Now, years ago, Marie Smith, who was a student in my lab, performed these experiments where he made reaching movements with this little robotic arm here. And you see these movements that he made, nice and straight to the target. And then he looked at individuals that have cerebellar degeneration. And what he noticed is just like what Gordon Holmes was saying, look, the movements start out okay, but then they have trouble stopping right on the target, as you see here. And we can see that also in saccadic eye movements, which I will show you with this video that I borrowed from my colleague David Z. So as you see, the movements are made, but the saccades do not stop on the target. They overshoot in this case, they overshoot, and so forth. And just you can imagine what a debilitating condition it must be to be able to just read a book when every saccade that you make is dysmetric. So the question that I want to come to is that what does the cerebellum compute that's so essential for ending these movements, Gordon Holmes's idea? And to do that, what I need to do is briefly summarize for you a little bit about the anatomy of the saccadic system. We know that there are direct projections from the retina to the superior colliculus. So the superior colliculus has the machinery underneath it to make saccades. It has projections to burst generators, which then project to oculomotor neurons that allow the colliculus to make saccades. But under normal conditions, the colliculus is not allowed to make saccades. Indeed, what happens is that that same information from the retina through the thalamus gets to the cortex and gets evaluated by our cerebral cortex, by the basal ganglia. The basal ganglia is inhibiting the superior colliculus, the frontal eye field, the LIP are exciting it, and through evaluating the stimuli around us, our cortex assigns utility to what's available, and then it decides, if it wants to make movement. And it increases its excitation to the colliculus, it removes the inhibition for using the basal ganglia, and then you make a movement. Now, the cerebellum sits on the side loop here. It gets a copy of this activity that's going on via the pons, and then it does some magical computation that this rest of the structure absolutely needs to make that movement well. Now, saccades have been studied for more than 50 years. We have absolute giants in the field who have studied these questions. David Robinson, Bob Wurtz, David Sparks, Okehide, Paul Glimcher, Mickey Goldberg, Richard Anderson, and Fuchs, Kojima, Sutidju, Thier, Z, just to name a very few people. And you might ask yourself, after all these years, Reza, who the heck are you to tell us anything new about this structure? And what I'm going to show you is that the basic problem that has remained is that when we measure neural activity in the cerebellum during the simple goal-directed movement, during a saccade, we really have a hard time understanding what is this prediction that it's making that's so important for stopping the movement on target. Okay, so to illustrate these problems to you, I just want to show you some recordings from marmosets. So marmosets are these small primates that live in the Amazon jungle. And because they're small, they have the advantage that we can use some of the modern techniques that were developed for rodents and record from many Purkinje cells and analyze their data. Now, the other important thing about marmosets is that they're primates, which means that they have a fovea, which means they make saccades. And now, as you will see, the statistical analyses that we will rely on to understand the language of prediction is going to require a lot of movements, and we're going to need multiple Purkinje cells are simultaneously recorded to make sense of things. And so for this reason, I think they're a particularly good model for studying the cerebellum. Okay, so let's look at some Purkinje cells of marmosets during movements. So here's a saccade that's made to the left. This is the velocity of the eye. Here's a saccade that's made to the right. Here's a Purkinje cell's activity. As you see that it's active before the movement begins, and it suppresses activity, and then it keeps on suppressing long after the movement is over. The idea being that when you look at an individual Purkinje cell, it's obviously cares about a saccade, it's modulated by the saccade, but there's nothing special happening near the end of the movement that you would say, okay, well, this is something that's being predicted about how to stop this movement. Other Purkinje cells do things that are even more complicated. Some pause, some burst pause, some burst in both directions, double burst in other directions, and whatever variety you. What's important is that individual Purkinje cells really don't make a whole lot of sense to us when we look at their activity from the perspective of how is it making a computation regarding a movement. Particularly, what is it predicting about stopping and so forth. So let's now get back to the learning problem. As I tried to describe it to you, we need to understand the cerebellum both from the perspective of control and learning. And so now let's bring together what can be used from a learning perspective. So suppose you have a three-layer network. In this three-layer network, input arrives and the output is the prediction of this network. Now, why do I bring up three-layer networks? Well, because the cerebellum is sort of like a three-layer network. Inputs come in using mossy fibers, computations are performed, Purkinje cells are the output of the cerebellar cortex, and finally the nucleus neurons makes the final prediction that becomes the output of the cerebellum. In a three-layer network, if you ever took a machine learning course, what they teach you is that to learn efficiently, what this network has to do is that every neuron that's going to contribute to this prediction that this neuron is making has to receive error information in order for it to improve the output of the network. So the way machine learning works is that you take the prediction that the network is making, you compare it to an observation, and then the result is the prediction error that is then fed back to the circuitry. Well, the cerebellum has something actually not that different. We also have output from specialized neurons that go to the inferior olive, and a comparison potentially is made here that then becomes this prediction error that goes to this network. Now, what's interesting is that in the cerebellum, this structure here, the prediction error when it's the climbing fiber input to the Purkinje cells is incredibly strong, whereas the input to the nucleus cells seems to be rather weak. So that's kind of puzzling. Why would this error information only go very strongly to Purkinje cells, but not to nucleus cells? If you were building a three-layer network, you certainly wouldn't design things then. And like most things in biology, a design like this suggests that there may be something to do with evolution. Maybe the cerebellar cortex evolved first and the nuclei came later. And indeed, in some primitive animals, we're missing the nucleus layer in the cerebellum-like structures that exist. So whatever the reason is, there seems to be a difference between these two pathways where error is coming. So let me show you that with some data. So here's our three-layer network, and here's our Purkinje cell. And I want to show you activity in this Purkinje cell. So you see two kinds of spikes. You see what are called simple spikes, just the regular spikes that are going downward here. This is the simple spikes of the Purkinje cell. And then there's these complex spikes. Complex spikes were named by Tom Tatch. Tom was a student in Hubel and Wiesel's laboratory, and they were studying the visual cortex. And, you know, in the visual cortex, he was influenced by the terminology that was being used, and he used the term complex, like the same way Hubel and Wiesel were using it to describe different receptive field of the cell. So complex spikes are inputs from the inferior olive that are projected to the Purkinje cell, providing potentially error information. You see it is incredibly powerful input that's coming to the Purkinje cell, potentially telling it, look, there was an error that was made in some form. The simple spikes are occurring at around 50 to 100 hertz. These are potentially the predictions that the Purkinje cell is making. Now let's look at what happens when one looks at the nucleus cell. This is data from Jaeger's lab. And what you see is that a optogenetic stimulation of the inferior olive produces only a tiny change in the membrane potential of the nucleus cells. So that same error information is very strongly going to Purkinje cells, but not to the nucleus cell. All right. So in principle, however, we need to have this network learn. And if we're to learn, we don't want just the middle layer to learn, everything has to learn. So what might be the needed aspect of this? We know from anatomical principles, from the work of Adi Person and Indira Raman, that there are about 50 Purkinje cells that converge onto a nucleus neuron. These are the parents of this child. So this child is making a prediction. That prediction is going to be compared to an observation, and there's this error. This error is going to go back. And the idea is that the error of the child is going to be conveyed to its parents. The complex spike is the teacher. Now, what we want to do is that if the parents all receive the same error signal from the olive, that may produce a synchronized complex spike in their output. And potentially that synchrony might be the teacher for the nucleus neuron. So the idea is that what we want to do is teach both the middle layer and the output layer. And if we wanted to do that, then we might organize these Purkinje cells into populations that share the same teacher, basically provide the same input, same teaching input, or a similar teaching input at least, to the parents, and then make the parents be the teacher for the child. So the idea is to understand the language of simple spikes. Might be useful to organize the P cells into populations that share a similar preference for error via their complex spike. So to understand the prediction, organize them first based on a preference for error, then see as a group, what are they predicting? So now, what is this error that Reza, you're talking about? What kind of errors are we talking about? So we're talking about saccades. So we're going to be describing what might an error be from the perspective of the superior colliculus. Suppose you have a monkey that sits in a room and is fixating a dot on the screen. The act of fixation requires activity in a region of the colliculus called the rostral pole. These are the regions that care about information around the fovea. In order to hold still, both rostral regions of the superior colliculus have to have neurons that are active. Now, suppose I show you a target to the right. That means there's going to be activity on the left colliculus associated with the onset of this salient stimulus. If your brain decides to make a saccade to that stimulus, then what happens is that activity in the rostral pole falls, activity in the caudal regions representing that stimulus rises, you make a saccade. As the eye ends at the target, arrives at the target, you expect now what happens is that the activity in the rostral region should go up, but this target has been moved. It's over here. So now you get a new region of the colliculus that's active. So this is the expected sensory consequence of saccades. These neurons should become activated because the stimulus should be on the fovea. This is the actual sensory consequence. That's the error. Error produces unexpected activity among neurons in a specific region of the colliculus. And that is going to then activate the inferior olive. That's going to produce complex spikes in our Purkinje cells. So what we're going to try to do is use error information to map the visual space of the superior colliculus onto the Purkinje cells. Find the cells that care about the same error, organize them into populations, and ask what are they predicting as a population. So this is a two-step process. The first step is measure how each Purkinje cell responds to prediction error. Those are the complex spikes. So here's our subject. She's sitting in a dark room. She sees a light on the screen. That's a fixation point. She sees a target. She makes the saccade to the target. We jump the target. So it's now producing a sensory prediction error. It's a different location than the fovea. Suppose it's to the left. Here's the data associated with some trials when the stimulus jumped to the left. The blue dots are the simple spikes. The red ones are the complex spikes. So you see that when the primary saccade ends, so the primary saccade ends at time 0, here are the simple spikes. Afterwards, there's a few complex spikes when the target jumped to the left. When it jumps to the right, there are fewer complex spikes. So what we can do is count the complex spikes here. The probability of a complex spike we see is different depending on where the error was. We can compute a tuning function of error for this Purkinje cell, and you see that this Purkinje cell responds to leftward visual errors, which means that there's a part of the superior colliculus that this Purkinje cell is receiving climbing fiber input about. Now, You can plot the timing of this complex spike response, and it's really interesting, this double humped response increase when the stimulus is in direction CS on. It's in the opposite direction, CS plus 180, you see it as suppression, which is also very interesting. Why do the complex spikes suppress? What's the information that's causing that? It's one of the mysteries that I don't have an answer for. Okay, but what this does is that it provides us with a preference for error. We know the direction that the error that matters. Okay, so now step 2, we're going to use that error information to define a new coordinate system for this Purkinje cell. What do you mean by that? So suppose you record from a group of Purkinje cells. So here's some Purkinje cells and you've recorded from them. This is the right side of the vermis, and you see there's some cells that care about upward errors, some cells that care about left, some cells about bottom left, and so forth. So what you do is that for every Purkinje cell, use the CS on vector, the direction of the preferred error, to now define a new coordinate system. So suppose you have a Purkinje cell, and this is the preferred error that it has. It cares about this error. But you've defined your screen coordinates like this. You say 0's to the right, 90 degrees up. Now, what we're going to do is we're going to make a saccade. Suppose the saccade is to this 45-degree angle. What we're now going to do is we're going to rotate this coordinate system. So 0 is its preferred error. So now this saccade is not towards 0, but is toward CS plus 250. So what have we done is to say, suppose that this cell has a particular region of the colliculus that it cares about. What we're going to do is we're going to represent the saccade that's actually made, not with respect to the screen, but with respect to this error, which is in this case, the visual space that is being mapped onto it. So we're going to throw away Cartesian coordinates. We're going to care about error coordinates, which in this case is CS on. Okay, to remind you, here was our puzzle. The animal made a saccade. Some cells burst, some cells pause, some cells do other things. There's nothing special about ending of the movement in individual Purkinje cells. Now what we're going to do is we're going to look at the activity of the cells and simply ask, when a saccade is made in a particular direction with respect to the preferred error, and add up the spikes, what do we get as a population? So the direction of the movement now is no longer in Cartesian coordinates, it's in CS plus 180. So some cells burst, some cells pause, but now you simply add the spikes of all the cells without weighing them, simply add them, and you see something rather remarkable. You see a burst followed by a pause, and it ends at the end of the saccade. It's telling you that without any magical math, we simply by finding the coordinate system of the visual space, mapping it onto the cerebellum, we get something interesting with respect to the population activity of the Purkinje cells. Now, if you look at other directions, you see that this coding is going away. So whereas in direction CS plus 180, you see this burst pause, the burst becomes smaller as the movement moves toward direction CS on, and the pause disappears. So as a population, we see this idea that there is this encoding of kinematics. As the movement is being made, the burst becomes larger as the velocity becomes larger, but the pause remains time locked to the onset of deceleration. So the burst grows with the velocity, the pause remains locked to the onset of deceleration. So what that tells you is that as a population, the P cells are predicting when the movement should be stopped, even though no single Purkinje cell gives us that data by itself. Okay, let me summarize where we are. How should we organize the cerebellar neurons into populations? What we did is that we imagined that the inferior all-level was like a city architect, planning the Purkinje cells into anatomical neighborhoods, producing populations that had a common preference for air. When the Purkinje cells in the vermis were organized based on this idea, their simple spikes appeared to predict features of the ongoing saccade, particularly the timing of deceleration. Okay, so Masao Ito showed us that Purkinje cells are inhibitory neurons. And it's really pretty difficult if you imagine now. How are you going to control the output of the cerebellum, which is at the nucleus, via inhibition? Because all you got here is inhibitory activity. But really what you want is to engage the nucleus neurons to do something. Presumably, engage the downstream motor structures, stop the movement when they're supposed to. So what I've shown you so far is that Changes in firing rates. I showed you that there is this increase in activity, then a removal of inhibition, the disinhibition, which potentially has something to do with stopping the movement. But there's another interesting aspect of this, and that has to do with synchrony of the simple spikes. And this is now really the power of the marmosets, allowing us to record simultaneously for multiple Purkinje cells and ask, is there something about the timing of these spikes that's also carrying information? And in order to show you that, I need to briefly summarize Abby Person and Indira Raman's work associated with synchrony. So 8 years ago, in a remarkable paper, what they showed us is that if one takes a group of Purkinje cells that are connected to a nucleus neuron and looks at their firing rate, so suppose that we have 8 Purkinje cells. Each of them are firing at 50 hertz. However, the spikes among these 8 neurons are no more synchronized than chance. So you just having this barrage of inhibition that's coming on this nucleus neuron. Well, the nucleus neurons is basically suppressed, it's not doing anything. It just hardly has any spikes. But now you take that same 8 cells and have them fire on average, again at 50 hertz, but make half those spikes synchronized among them. Now you see something remarkable. You see that the nucleus neuron begins to fire. It appears that by synchronizing their spikes, they get to entrain the nucleus neuron, making it fire. So this raises the question, do Purkinje cells synchronize their simple spikes during movements? And the answer is remarkably yes. But what's really amazing is that they don't synchronize their spikes when there's this burst in their firing, but they synchronize it when they are suppressing their firing near end of the movement. And again, that synchrony seems to wane as the movement direction changes. So it appears that synchronization of the simple spikes is greatest during deceleration of the saccades for which there was disinhibition, suggesting that there's coordination of disinhibition, the firing rates, and synchrony in order to force the nucleus neuron to fire. Okay, to summarize, how might these Purkinje cells take their inhibitory output and get the nucleus neurons to fire? What we saw was that Purkinje cells that share a common preference for air as a population had this firing rate modulation that seemed to be maximum during deceleration. However, they also synchronize their spike during deceleration. So maybe by both reducing their inhibition and synchronizing their spikes, they're able to drive nuclear output. All right, so what's this nuclear cell doing with this input that it's getting? How is it relevant for stopping this movement? Basically, what we need to do is ask the question, what's the relationship between the error information that the Purkinje cells get and the downstream effect of the nucleus neuron that they project onto? Is there such a relationship? So here's the activity again. There's a burst followed by a pause. There's this increase in synchronization. And now we have this question, when we have a group of Purkinje cells that have this preference for error, is there a relationship to this nucleus neuron's projection? And there's some ideas about this, and indeed some data that suggests that there is this consistent relationship between a Purkinje cell's response field in terms of complex spikes and the direction of the action of the nucleus neuron it projects. So the idea is that if a group of cells prefer this error, then they project onto a nucleus neuron that does something to relieve that error. So they don't just go anywhere. They go to a nucleus neuron that has a consequence in actually doing something about the error that these Purkinje cells care about. So this allows us to ask a question. When these Purkinje cells reduce their activity below baseline and synchronize their spikes, are they getting some behavioral consequence that's associated with stopping this movement? So how are we going to ask this question? This is the work of my student Solomon Mueller, who has been analyzing our data, and this is what he found. So what Solomon did is that he looked at a single Purkinje cell and said, look, these are the simple spikes of this single Purkinje cell. Here's when the saccade takes place. In this case, this single Purkinje cell is bursting. Now, if you look here, there are no complex spikes, and you see occasionally a complex spike here and here. These are little red dots. Then what Solomon did is that he organized your plot here so that you have a complex spike from this point on as a function of time. And what you see is that there's this suppression of the simple spikes for about 5 to 10 milliseconds. after the complex spike. So the complex spike is such a powerful input to the Purkinje cells that it suppresses production of simple spikes. And when you think about it, it's really a brilliant system for engineering analysis, because complex spikes are perturbing the ability of the Purkinje cell to produce simple spikes. Does it alter the movement? And if so, what's the consequence of that complex spike in altering this movement? And so this allows us to ask the question, do natural perturbations that arrive via complex spike have a consequence on behavior? Remember, complex spikes suppress the simple spikes. So now we can ask the question, does the simple spike suppression produce a change in behavior that has anything to do with the preferred error of that Purkinje cell? And the answer is as follows. So you take the population of neurons. This is about 270 Purkinje cells. You ask, on a single trial, there was a complex spike during a specific period of time. Here it is. And so during this period, there's a complex spike in one Purkinje cell. So of course, the simple spikes as a population get slightly reduced. What's the consequence of this on behavior? If you compare a saccade where there was a complex spike followed by suppression of simple spike in a single Purkinje cell to the same saccade made at a different time where that single Purkinje cell did not have the complex spike, what's the difference in behavior? And what's really remarkable is that the complex spike, if it suppresses, when it suppresses the simple spike, it doesn't change eye position in direction CS plus 90. It pushes the eye in direction CSI. So suppression of the simple spikes during a saccade pushes the eye just slightly in direction CS on. Look at the y-axis here. 5 to 6 degrees per second. A saccade is 600 degrees per second. So this is 2 orders of magnitude smaller than the movement itself. But nevertheless, because of spike triggered averaging, you can see the influence of that suppression of the simple spike. So simple spike suppression during a saccade leads to force production in direction CS on. What does this mean? Now we have a way to understand how the cerebellum might stop a saccade. So we have a group of Purkinje cells that we imagined receive climbing fiber inputs that organizes them so that they all prefer these errors in the same direction. When there's suppression of simple spikes, we saw that it causes behavioral responses that push the eye in the same direction as the error. So now, if you take my activity, here's the burst followed by a pause in direction CS plus 80. So during this period, when I'm pausing, I'm producing forces in direction CS on, which is opposite of this movement. Thus, this is likely in training the nucleus neuron, producing forces in direction CS on, thus stopping them. So we now have a mechanism to understand how the suppression followed by synchronization is influencing downstream neuron, which then is going to go and activate presumably a behavioral consequence that will oppose this movement, stopping that movement. All right, let me stop for a second and ask a different kind of question now. On Wednesdays in the evening, I go and visit my mom. This is my mom. And I talk about some of the research, and she's interested in this stuff. And she asked me, what are you working on, Reza? And I talk to her. And frequently she tells me, so what? You know what? You're discovering this and that, but what does this mean? And what does she mean, so what? So at the beginning of the lecture, I showed you this individual who was suffering from cerebellum disease. And you know what she's asking is that if this person was watching this lecture and heard your results, wouldn't he want to know, what good is this? What your understanding of what this stuff is about? And now, to be honest with you, most of my life I have not cared about this question, but my mom makes me think about this. And so I thought I'd present some ideas about this for you. So this is the diagram that I've been showing with regard to the mossy fiber input and the 3-layer structure. And so you might imagine that cerebellar disease is killing these Purkinje cells. And you might ask yourself, okay, now that you understand something about the cerebellum, how can this be of use? And what I'm thinking is the following. Wouldn't it be nice if we had a circuitry that some way decades from now we would insert into the cerebellum that would listen to these mossy fiber inputs and produce the right output to the nucleus cells. And effectively, like a deep brain stimulator that we have for Parkinson's disease, much more sophisticated one might exist for the cerebellum. So I've been dreaming about this, wondering what this might be like. And I want to show you some results based on this idea. So with my student Amin Fakharian, we can record from the mossy fibers that arrive into the cerebellum and provide information. And I want to show you what is the input to the cerebellum, because in order to build this emulator that I just described to you, we need to be able to understand what the input is and provide the right output. So it turns out that the mossy fiber input really produces a very nice representation of the visual space. So for example, here's a mossy fiber input during for saccades that are made in various directions. So you notice that this input cares about saccades at the top left, but this map here shows you it doesn't just care about all saccades, actually cares about a specific amplitude saccade. So it looks like a colliculus representation of the visual input. It's saying this is the movement that I'm about to make, but I have a response field associated region of this movement. We also have cells that look like this. This provides input that doesn't have a response field to say this movement, but it looks like motor commands that are being copied and sent to the cerebellum. Here, what you have, activity that grows as the magnitude of the saccade increases, and it looks like basically you have a copy of what you might imagine to be the burst generators that are producing information about, where the movement should take place. About half of the mossy fiber inputs look like this. But then we also have cells that look like this. These are pausing regardless of the direction of the movement. And you might imagine that this is input from either the rostral pole of the colliculus, potentially that, those regions that I described to you that care about fixation, or maybe from the omnipause region. So this is roughly the input to the cerebellum. All right. So my mossy fibers look like this. This is the input that's being provided. I have in some of the mossy fibers information about where I'm supposed to go, the desired displacement of this movement. In other mossy fibers, I have efference copy. I have information about the motor commands that are being generated. Now, suppose this is the two pieces of information that you have. Now, what we want to do is say, if this is the input to the cerebellum, how do we get to the Purkinje cells that I showed you as a population, predicting when the movement should be stopped? So what would you have to do to go from desired displacement and a real-time estimate of efference copy to tell me when I should stop the movement? Well, what you would do is that you would integrate the inputs that are efference copy, make predictions about the state of the eye. That would be your forward model. X hat would be your estimated displacement. You've taken efference copy, you've integrated, added it, in this case, you come up with an estimate of how far the eye has gone, you compare it to the desired displacement, and until it hasn't reached it, you don't do anything. When it reaches it, you suppress the Purkinje cells below baseline. That becomes the output of that system. So what this looks to me is an integration to bound. You have cerebellar cortex that's integrating input from one type of mossy fibers, that's the desired displacement, to a bound, that's the motor commands, to a bound set by another type of mossy fiber. That would be the desired displacement. So you're taking efference copy, you're integrating it to estimate displacement, you're comparing it to the desired displacement. When the two match, you suppress the Purkinje cells. That becomes your prediction about when to stop the movement. So you both have a forward model that makes prediction about sensory consequences, but you're not just stopping there. You're comparing your sensory consequences to the desired one. When the two match, you stop the system. Okay, so this is a fantasy. I don't know if that's true, but the idea is that if this was something close to reality, then maybe someday down the road, we might be able to build an emulator that does take the mossy fiber input and then provides, because it has the information it needs about this particular movement, to provide the output it needs to the nucleus cell. So I'm going to stop in a few minutes, but before I do, I want to highlight for you some problems in the story that I've been telling you. The first problem is synchrony of complex spikes. So remember, I began my fantasy of how to put together Purkinje cells into populations by saying that the parents need to teach the child, and that might happen if there's complex spike synchrony. But I didn't show you anything about complex spike synchrony. So what's that about, Reza? Is that not true? Well, it turns out that there is some truth to that. And if you go to SFN, JPI is going to show you data regarding complex spike synchrony. So there is something about complex spike synchrony that is also not far from the truth. The second problem, what I've been showing you, is that this concept of integration to bound, the idea that you have a forward model that integrates and then you compare it to the goal and then you stop the movement when it gets there, it requires neurons that would actually do that. You would have to have some kind of integrator in place in the cerebellar cortex. And my student, Amin Fakharian, is going to present a poster at SFN that's going to show you data regarding this idea. There also seems to be some truth to this issue. But there are plenty of problems that I haven't really addressed. One of the most interesting aspects of it is that the problem of learning. So if you believe the data that I've shown you regarding synchrony, then what you're seeing is that the concept of prediction in the cerebellum comes both from the firing rate modulation and from synchrony, which means that when there's an error, it needs to not just change the firing rates on average, it needs to organize the Purkinje cells so the timing of when they spike become better aligned or less aligned. So synchrony must be a parameter that can be learned by the cerebellar cortex. And how the heck does that happen? I really have no idea. And finally, I've been talking about Purkinje cells. And it's important to realize Purkinje cells are not the output of the cerebellum, right? That's just the middle layer. So what about the output? Well, that's the nucleus cells. Why haven't you talked about the nucleus cells? Because I don't understand the language of the nucleus neurons. I don't know how to put them into populations. I don't know what the output in cerebellum really is. So that remains a major puzzle that I'm hoping that again, with our marmosets, we'll be able to make some sense of it. All right, I'm going to finish now. So we talked about population coding in the cerebellum, and we use this idea to put cells together based on the concept of preference for error, using this machine learning based notion of, well, you're going to have the parents teach the child, and the only way for that to happen is that the parents all get consistent information about error. When we did that, when we organized populations based on a preference for error, what we saw was that the simple spikes made some sense. First of all, they end when the movement ends. Then they show this burst and a pause, and the pause happens during deceleration, or the disinhibition, I should say, occurs during deceleration, and there is increased synchrony among the neurons, all of which really suggests that it's an attempt by the Purkinje cells to drive the nucleus neurons, get it to fire. And I showed you that if that happens, there is a force production in the direction opposite to the movement, which would then allow it to stop. I described a potential way to think about in general the computations that are being performed. What we see in the mossy fibers is two different kinds of information: the desired goal of the movement and the motor commands that are being produced in real time. And if one were to integrate these motor commands, integrate in time, you would have a forward model that would predict the state of the eye. There would be some kind of a comparison between the state of the eye and the desired state. That would then result perhaps in this stopping the movement as the Purkinje cells disinhibit the nucleus. Okay, I'm going to finish with a much more beautiful way to express movements than I have been able to do for you, and that's by Hafez. He's a 14th century Persian poet. Here's what he says about motor control. He says, because of our wisdom, we will travel far for love, as all movement is a sign of thirst, and speaking really says, I'm hungry to know you. All right. Thank you so much, and I'm happy to answer any questions for you.
Reza.
Yes, sir.
Quick question. That most cerebellar patients overshoot for saccades coming back towards the center of gaze, and undershoot, which means that they're stopped prematurely going out. And I've always thought that's an issue of compensation for elastic restoring forces. So, it's not only the issue of stopping erroneously
Right.
in the Gordon Holmes sense.
Yeah, the work of Roby and Yoshiko and the Fuchs lab and others have shown that the damage to the nucleus produces dysmetria that is hypo in one direction, hyper in the
Right.
other direction. And what we see with our Purkinje cells is that in direction CS plus 180, they show the suppression, but not in direction CS on. And we do see this separation. This basically cells have response fields for error to the opposite side. So right side of the vermis has leftward preferences. So if the way I'm thinking is that basically if damage is asymmetric, it's going to produce. So if there's one side has greater damage than the other side, it's going to produce largely damage associated with the preferred errors of those cells. It's interesting that I don't know if there's global damage. I guess that we just lose the ability to predict when to stop in all directions. But if it's unilateral, they tend to be for the ipsilateral direction.
But we see that kind of directionally position-dependent dysmetria in patients with generalized cerebellar disease. I mean, the vermis is not blessed like with an end artery, and it's almost impossible to get a half vermis. I've never seen a half vermis lesion.
Yeah. No, you're right. Yeah. And then we're still far from the nucleus, right? So we don't know what precisely is the output of that circuitry. Yeah. Let me go to some people who have raised their hand. I have a hard time seeing who that is. So just go ahead, and if you raise your hand, please go ahead and jump in and ask the question.
Hi, Reza. Can you hear me?
Yeah. Hi, Dan.
Hi. Beautiful talk. I had two questions. One is maybe more specific, and the other is more conceptual. The first is, I wonder whether if the motor command that's coming in through the mossy fibers is somewhat predictable over time. I guess the question is, do you actually need integration? In the simplest case, if it were just a static amplitude, then you could just multiply by something to predict what the end is. Do you actually need to do integration over time, or is it something where the cerebellum could predict the endpoint?
Yeah, so I think you do, and I'll give you the reason for it. So if you look at the mossy fiber inputs that we think represent the motor commands, they are active for both high vigor saccades, low vigor saccades, targeted saccades, spontaneous saccades. They're just an exquisite representation of these motor commands. And what happens is that for some 10-degree saccades, you move slow because arousal is lower. There is no visual stimulus there. You're just making a saccade in the dark or spontaneous saccade. Or you're making it at the beginning of the experiment where arousal is high. This is a rewarding stimulus. You have up to 10 to 20% greater velocity. And that precision requires integration of those commands. So we don't know if that integration takes place. We don't know if there are cells that integrate, but we're just imagining that if you wanted to have a forward model that predicted this, that's what you would need.
Got it.
Thank you so much.
Can I ask the conceptual question?
Sure.
Maybe this is naive. I'm just wondering whether if you were to show the same sort of error to the marmoset over and over again, do you have a sense of what the persistent effects of this are in terms of the simple spike coding? Right. You mentioned learning, but what is the persistence?
Excellent question, Dan. So Jay's work, he finds that there's really a big difference between measuring complex spike response in terms of firing rates versus measuring its synchrony. We think that error really is about synchrony, not about firing rates. So there are many things that cause changes in complex spike firing rates, stimuli that are predictable, movements, not just errors, but just events that activate the superior colliculus. So we don't think of superior colliculus as an error system. And if the superior colliculus maps onto the inferior olive, we can't really imagine inferior olive to be an error system either. But what we see is that inferior olive, the complex spike, are much more synchronous when that's an actual error in the movement versus just a sensory event that resulted in a movement. So it's a long ways to go to understand the difference between these variables. But I suspect that understanding of the cerebellum is really going to require this language of timing as well as firing rates. Next question. Hi.
Hi, that really was a
Hi, Jerry.
wonderful talk. So
Good
as
to have
someone who studies development, I'm probably going to ask you a question that is impossible to know, but I would welcome speculation if you feel comfortable doing it. This is all based on this concept of what is an error and what is the correct movement. And, you know, as this system is developing, how do you think it learns what the intended consequences of a movement are and how to define an error? And like, how does the cerebellum really get these models up and running?
Right. One of the key things, this relates to some of the new work that Mickey Goldberg has been doing. And you know, many kinds of learning do not have an obvious relationship between error and correction. You have to figure that out. And one of the things we see in complex spike response is that all kinds of events activate the complex spike. So the same way as the colliculus being this multimodal structure that responds to visual, auditory movements, or perceptive even, that many people have found that climbing fibers respond to many things. And perhaps there's a modality that they specialize in, but they will indeed respond to many things. So I think in principle, the visual motor system is being reflected into the climbing fiber input onto the Purkinje cells. Some of those events are indeed what we would call errors. Others are just events that are taking place. And how does that then result in learning? What are the consequences of it? It's you know, Jimmy, it's honestly such a beautiful question, but I think people that have been studying tasks that are a little bit richer than the one that I've described to you, like the work of Mickey, they're really pushing this boundary because, you know, in those scenarios, there isn't an obvious relationship between error and correction, but yet the animal learns and it still requires a cerebellum. So how's that taking place? It's a good direction to go.
Thank you very much.
Hi, Reza.
Hi.
Yeah, thanks so much for the great talk. I really enjoyed it. So my question is, I mean, we study the microcircuits of performance monitoring in the cortex. And so I was wondering, to what degree do you, now you talked about motor errors, but to what degree do you think what you described can be generalized to other sorts of error detection? And whether the circuit motifs that you described can be generalized across the cortex as well?
Wow, that's a very tough question. I mean, I think let me turn this over to you and ask, well, you studied the cortex, and what is there something that you saw here that you thought might be relevant to what you studied?
I mean, a lot of the way the way we interpret our data in general is inspired by these kinds of models, because the understanding of the motor circuitry is better than some of these cognitive areas that we study. And so, but I just wanted to see what your general idea is about the role of cerebellum, maybe with respect to other modes of error detection, performance monitoring, and
let me admit my ignorance first to say that's a large field that you're asking me to make a comment about, and I can tell you that the state of the animal. For example, recently my student Paul has been looking at the state of the animal as measured via the arousal state, say via pupil diameter, and we see these really beautiful correlations between the arousal state of the animal and the vigor with which they make movements. And we've measured the climbing fiber response, the concept of error, and I'm really itching to know, is the arousal system modulating the strength with which the error information is coming into the cerebellum? And from a perspective of the colliculus, I think that the data regarding reward suggests that vigor of movements are coincident with changes in firing rates in the colliculus, which may then alter what I've been calling error and how it arrives into the cerebellum. And finally, the whole concept of integration to bound, of course, is a cortical idea. It's an idea that's reflected in decision making. And so I've borrowed this idea and I've proposed it that perhaps computation like that is taking place in the cerebellum, but that it really remains to be seen.
Okay, thank you.
Hi, Reza.
Hi, Mehrdad, good to see you.
Super fun talk. Thank you. It's like a master class in cerebellum. It was great. Quick question. If you were to speculate, a lot of people have indirect, peripheral evidence that cerebellum is involved in timing. If you were to speculate of how this formulation will generalize to errors for timing in the cerebellum, would you then predict that you will have like time cells in terms of complex spikes coding in the cerebellum, or I guess, however, what do you see as the error being represented by the Purkinje cells in a timing task?
Yeah, that's such a beautiful question, Mehrdad. You've done such lovely work in looking at the learning of timing, and the classic way of thinking about timing, I suppose, is a classical conditioning where a stimulus arrives. Sometime later, something is going to happen, and you're going to have to predict the timing, and in this case, close your eyelid. And the way I think about it is that that's a learning to associate a future event to this condition, to be conditioned stimulus, and you have to count time, which would be simple, again, another form of integration. In this case, the bound is set by the climbing fibers that taught you when that event would take place. So in principle, I think learning to make predictions about time are effectively learning about these events that need to be counted. Now, one of the really remarkable things we see in the cerebellum are these metronome cells. I don't know what they are, both in the nucleus and in the cerebellar cortex. There are these cells that if I didn't know better, I would have thought that we've just mistakenly taken a MATLAB code and just generated these frequencies, because they are so precise in timing intervals. And perhaps they're these unipolar brush cells. I don't know. But again, it suggests to me that, yeah, you have this potentially the circuitry in place specialized for some form of integration, either be, of course, integration is a time-dependent process of adding until some event has taken place, and then you predict your output.
So I guess if we go, the rhythmic cells are, I don't know how to fit into the story that you told us about the saccades. But if I were just to extrapolate from the story you told us about saccades, let's say an animal wants to produce a time, be it maybe a conditioning, whatever it is, after a stimulus. Would I be correct to say that the generalization of your formulation means that after that stimulus, there will be Purkinje neurons that will be tuned to different time, giving us complex spikes at different times relative to some threshold, and that will be the way the error will be encoded?
The way I'm thinking is that when the event comes, the error produces complex spikes. The way perhaps we would then replace this input, not from you, but this would be internally generated. It would be those metronome cells that I mentioned to you. It would be addition of those metronome cells that then produce basically a counting of time, if you will. But again, I've almost never seen individual Purkinje cells do anything like this. This just comes out of the population. Your question is about how would the error then organize this circuitry so it could count like that? And boy, that is a really fantastic question, because I haven't talked at all about how error produces learning here, right? It's just been about this intrinsic relationship between the complex spike response and the Purkinje cells. But in your condition, you would have to learn the timing. And how that takes place is really wide open for me.
Okay, thanks.
Well, we're at our time limit, everyone. Thank you so much for your time today. I hope this was of some use. I appreciate your feedback, and I wish you a wonderful Tuesday. See you later.